Hello, everyone. My name is Mark. I'm a PhD student from Professor Zimmel's group at Technical University of Munich. Today, I would like to present my project about developing switchable fluorescent light-up aptamers using Toho media strand displacement and Puring rebel switch elements. Fluorescent light-up aptamers are rapid fluorescent reporters compared to fluorescent proteins. It has great potentials to be applied to RNA-based genetic circuits. Therefore, developing novel switchable fluorescent aptamer, which have a high on-off ratio, would contribute not only the RNA circuits, but also in vitro RNA detection. In this project, we try to adopt a new strategy that combines the basic switching mechanism of rebel switches with Toho media strand displacement and modified Puring rebel switch aptamers. The natural rebel switches contain an anti and anti-RBS to regulate the accessibility of the downstream RBS sequence, which reduces the leakage and increases the dynamic range of switching to a large extent. Ligand binding initiates the secondary structural rearrangement of the rival switch, while the rational designed Toho switch uses trigger RNA to unwind the Toho hairpin through strand displacement process. We combined the anti-RBS switching principle with strand displacement, so we can use our trigger RNAs to either switch on or switch off or fluorescent light-up aptamer, that is spinach aptamer. Now, if we look at the RNA structure of the spinach aptamer, 12 critical nucleotides are responsible for forming the G quadruplex and the binding pocket of the fluorophore DFHB1. Similarly, the guanine and adenine aptamer also include 20 critical nucleotides. However, the P1 stem of each aptamer can be modified into different sequences to regulate the downstream RNA structure. Now, if we look at the design and results, firstly, we try to switch the spinach aptamer using Toho strand displacement. The upper part is the switchable fluorescent activator. When trigger RNA is absent, the anti-spinach sequence is base paired with partial sequence of the spinach aptamer, preventing the binding of the fluorophore. The trigger binding will release the anti-spinach and restore the spinach aptamer. The lower part is the spinach switchable repressor. When trigger is absent, anti-spinach is sequestered within the loop region of the Toho hairpin. The trigger RNA binding will unwind the hairpin and anti-spinach will sequester partial sequence of the spinach. Therefore, the fluorophore cannot bind anymore. In order to fine-tune the dynamic behavior of our spinach aptamer, we designed 8 different anti-spinach sequences and we test them in vitro, and some of them showed much higher on-off ratio compared to the previous works. We also try to use modified guanine and adenine aptamer to control the folding of spinach aptamer through the combination of the above anti-spinach sequence and the modified P1 stem. We tried to develop 2 activators and 2 repressors, which can be regulated by adding certain concentration of the ligands. The in vitro experiment result showed that the guanine aptamer both showed high on-off ratio. However, the adenine aptamer showed lower on-off ratio compared to the previous one due to the lower binding kinetic of the ligand. We also tried to extend the functionality of the Toho-based spinach aptamer. We added additional Toho hairpin, which contains second regulatory sequence to regulate the folding of the spinach aptamer. Firstly, we tried to combine 2 spinach activator structures and construct them into an AND gate RNA switch. Only when 2 triggers are present, the spinach aptamer can be restored. We also try to incorporate 2 spinach repressors. When any of the trigger is present, can repress the folding of spinach and exhibit NOR gate behavior. The in vitro results showed both of them worked and very good and clear. In addition, we also tried to combine the spinach activator and repressor and develop it into a not implied gate RNA. However, due to the fast binding kinetic of the fluorophore, the repression from the 3' end anti-spinach is limited, which generates a certain leaky signal when trigger A is present. In summary, we developed and characterized 28 switchable fluorescent light-up aptamers, which can be either turned on or turned off using trigger RNAs or small ligands. We design and characterize different anti-spinach sequences and riboswitch stem sequences to fine-tune the switching behaviors. And we also develop and characterize 3 logic gates, RNAs, to control by double toehold hairpin, which are regulated by 2 RNAs. Lastly, our toehold-based spinach activator has great potentials to be applied to in vitro RNA detection, especially fast corona test. I would like to thank all my sponsors and thank you for your attention. I'm glad to answer your further questions and discuss more details with you. Thank you very much.